Welcome to class 11 students. This is the continuation part of the chapter chemical constituents of living cell. In my previous video I completed discussing about all the biomolecules and their details. Details include classification, structure, functions, etc. I hope that part is clear to you all. Next, very important and big part of the chapter that we are going to talk about today is enzyme. Now you all know what enzymes are. You have more or less some idea about enzymes, but today we are not today, in this chapter we are going to learn how enzymes work, what are the types of enzymes, what are the different components of enzymes, their mode of action, etc. Okay, so to start with, what is the definition of enzyme? How can we define enzyme? It is an organic catalyst. Means catalyst meaning everybody knows any substance or any molecule that accelerates the rate of any reaction without itself getting affected, right? So enzyme is also like that, but it is an organic catalyst. Organic means present inside living cells. So an organic catalyst, usually protein in nature, produced within the living cell that accelerates a specific biochemical reaction by lowering the activation energy required for that reaction is known as enzyme. Here we come across another term which is known as activation energy. Sorry. I don't know why it's not coming here. Okay, so activation energy. Okay, so in this definition you came across a new term which you don't know, which you haven't heard before, is activation energy. Now what is activation energy? To learn that, you have to go to the first page where the enzyme topic starts. There you will find the definition of energy of activation of reaction, which is ultimately means in other terms it is known as activation energy. Now how is it defined? The energy utilized to activate the molecules taking part in the biochemical reaction is known as activation energy, or energy of activation of reaction. So from the name activation energy you can understand that it is some kind of energy required to activate something. So to activate what? In case of enzymes, here the molecules need to be activated, right, so that they can take part in the biochemical reactions taking place within the living cells. So here activation energy is the energy utilized or the energy needed, required to activate the molecules taking part in the biochemical reaction. Clear? Now if we read the definition of enzyme again, it will be clear to you all. An organic catalyst, usually protein in nature, produced within the living cell that accelerates a specific biochemical reaction by lowering the activation energy required for that reaction is known as an enzyme. I hope the definition of enzyme is clear to you all. Next we come to the naming of enzyme. How do we name the enzymes? You will find one thing very common in all the names of enzymes is that the names of enzymes, all the names of all enzymes end with ase, ASE. Okay, this is the ending of all the enzymes. So whenever you come across any word which ends with AAC, means any scientific term or a biological term which ends with S and you don't know whether that is an enzyme or not, just by looking at this AAC part you will get to know that it is an enzyme. Okay. Now, location of enzymes can be found in cytoplasm, mitochondria, lysosome, microsome, etc. These are all the locations of enzyme. Now we come to the chemical nature of enzyme. For that I am going to show you a slideshow. So these are the components of an enzyme, as you can see, but you have never come across this term hollow enzyme. So hollow enzyme, the word hollow means full or complete. So a complete enzyme is known as a hollow enzyme. Now what are the main components of an enzyme? It mainly consists of two parts, which one is known as the apoenzyme. The first one is known as the apoenzyme and the second one is known as the cofactor. Now what is the apoenzyme? The protein part of the enzyme, as you have learnt already in the definition that enzyme is proteinaceous in nature. So the protein part of the enzyme is the apoenzyme and it is basically the inactive part of the enzyme. Okay, and now the other component is known as the cofactor, also sometimes called as coenzyme. Okay, and that is the non protein part, as you can see it here, non protein part, and it actually acts as an activator. It activates the apoenzyme which is inactive. Now when we combine the apoenzyme and the cofactor, we get the hollow enzyme, which is basically the whole enzyme, and this hollow enzyme is active. Why is it active? Because when coenzyme, the activator, comes across the apoenzyme which is inactive, it activates the apoenzyme and then it starts to show its enzymatic activities. Okay. So in this picture you can see an enzyme, the enzyme here, and different sites of the enzyme. So this is the active site, this is the allosteric site, this is the substrate, and this is the any inhibitor, or it can be any inducer as well. So here my point is to explain to you what is allosteric site. Active site you will get to know later on, but my point is what is allosteric site. So the site or the place of the enzyme where any effector molecule, that can be any inducer or, as in this picture, inhibitor, they come and bind to the enzyme. That part of the enzyme is known as an allosteric site, and that enzyme is known as an allosteric enzyme. Now what is an allosteric enzyme? I previously told you that the allosteric enzyme can be either an inducer or an inhibitor, and due to the binding of this effector molecule to the enzyme, the configuration of the active site, the configuration of this place might change in favor or not in favor of the enzyme, and accordingly the substrate will either bind or not bind to the enzyme. Okay, so this is about allosteric enzyme, and the place where the allosteric enzyme binds with the enzyme is known as allosteric site, and this whole property is known as allosterism. I hope it is clear to you all.